Liverpool, UK. He's our intermediary from the Europe. So we recognize him in this program. So we have the female in our midst. Though they are not here, they are part of the admin, but these are the founding. So that is the members of the team of African Life Science RNA Salon of Biochemistry, Alex Ekome Federal University. Please, Doc, can you move forward to the high table? Thank you.
Our help is in the name of the Lord. Thank you, Almighty Father, Creator of heaven and earth, for this opportunity of having us together for this symposium. We are very grateful to you, O Lord, for granting all of us who are here and those who are on their way coming, joining mercy. We present the activities we are going to go through today into your heavenly hands. We ask you, O Lord, to direct the affairs of this symposium so that they key at the end of our deliberations. We commit our presenters into your hands, and we ask that you give them that leverage to enlighten us by your name, and we say that when we are through with the program our places, in Jesus' mighty name we have prayed. In the name of the Father, of the Son, and of the Holy Spirit.
I encourage you to try to see what you can do for our country. We all know that our country is going down, and I think it will continue to go down until we do something. We always pray for the God of Israel to do our chores for us, to do our things for us. But here is Israel, the same Israel. We fighting Hamas with the technology. The point now is, do we have the technology? Do we have what we can call technology? Better than I am. If I had access to information you have, I'm not better if I have access to technology you have access to. But all the same, we can do what you can do for our country, which is going down. Which is already down, not going down, it's already down. I welcome you to visit it. We are work partners in progress. We are trying to see how we can train our students to be better than what they are. I did a manual research, like I told you. We did our own research manually because we belong to old generation, analog age. And so I encourage our students to be better. In my experience and in my research, I discovered that the problem we have in Nigeria is lack of citizenship. What can you now really put your chest and say, I am a citizen of Nigeria? Can you now say that you belong to Nigeria? Can you do anything for the sake of Nigeria? Politicians are not showing us example. Whenever there is a little money for Nigeria, they take everything. So our Nigeria is a take-all Nigeria. When you have the opportunity, you take all. And we now discover that the only way out is for us to develop citizenship and align myself with the proprietor of this place, Reverend Father Emmanuel, who came up with a philosophical ideal with which we can develop citizenship. That's where we're going to start. And that is going to start in schools. He came up with a philosophical thought they call epitizing. Epitizing stresses the development of the individual. When I was following him, I discovered that it traced the origin of man, the etymology of the word man. How did man come? He traced the etymology from the Igbo concept of man. Mad or mando, but man, that is the good that is. That means man is created by the Supreme Being, who is good, the Father of all goodness. He created us to be good, but we deviated from being good to being something else. And I think we try to develop our students towards attaining the goodness of man, towards citizenship, so that when a man develops into being good, into something good, he can now aspire to be good. And if he aspires to be good, he will now think of us and not me. Us and not me. So it's not enough to develop technology. We must develop a technology that talk about us, not me. And so I welcome you. You started here. Everybody's speaker. Everybody's people. If you stay here for just a minute to be happy, because everybody will. You will be like one of us. I thank you. I welcome you, and I implore you to relax. Be happy. If you see any lecturer or any student here, talk to him. You are sure you are talking to your brother. You are sure you are talking to your sister, and we don't make bad words. Thank you very much. As we continue with our program, I all us, especially our students, will listen carefully and benefit from the program. It is for the future. When I read the philosophy of this program, I now know that I understood that it is for the future. And whatever we do now will be for the future. Very soon, the four walls of the classroom will fall down. It will collapse. Because education will no longer be what happens in the classroom. It can be done online. It can be online, and people will learn online, and people will learn skills, and people will learn things. These days, in our days, we cram a lot of theories, laws in our head. We have everything in our head. Equations. But these days we are not ready for that. What we need now is for you to develop a skill that make you function in the society. What is important now is for us to be better. For what education can do for us. Not well, I mean not say not we can do what we can do for education, but I want to say that if I go to school, for example, and I'm a building, and I'm going about hungry, looking for a job, then I'm a fool. I had a PhD. After that, I thought of what to do. I started doing something beyond academics. Even at the time, I told my brother that I want to retire because I want to go to the village. I go to the village. Look at my village is just on it. If you come to the you see a lot of illiterates doing a lot of things and making a lot of money, and we that are very educated are looking for jobs, hungry, and we are graduates. I want to join what they are doing with my knowledge. So we must look at education that will help us, and this program I see can help us for the future. It can help us if you get into it. And wait, get to the point where we get to benefit from it. For the organizers, I thank you. I say thank you very much for bringing to our doorstep. I want to, after that, I want you to organize something for our students. For our students on the so that our students can benefit from it, not just the students that are in biochemistry and so on and so forth. Thank you. Welcome.
Thank you. Can we put hands together for Prof. of Osatec Polytechnic Enugu, Professor Mike Ike Obudele. So today we will see Dr. Mrs. Antonia Nkiruka Mba, the head of Department of Microbiology and Parasitology, David Umar, Federal University, calling women to stand up for science in her own keynote presentation. You're welcome, Doc. So I will only tell Associate Professor Aja to keep on presenting, while those that are on Zoom should join Zoom and hear him, so that we'll quickly do that and proceed with our presentation. I want to assure you that we will not stay beyond 3. We must finish before that time. Thank you and God bless you. So let's wait patiently for him to begin.
for the inconveniences and all the technical issues. I wish you the best celebration. Thank you.
Thank you very much.
Thank you for your patience from Uganda. So we are proceeding immediately to the keynote presentation. We have our keynote presenter this morning, Associate Professor Ikechukwu Okore Igwenyi. He's an Associate Professor of Medical and Nutritional Biochemistry from Ebonyi State University. Prof is a big elephant. That is the NUG, ASU chairman of Ebonyi State chapter that is in your midst. And he is a vibrant scholar. He has so many programs, but he said, because I see you, Peter, because I see the team and I can see what you are doing and I want to be part, I will make out time, and he is here with us today. So let us welcome him to the podium. Thank you.
Director, the registrar, the organizers of this event, my colleagues, students, or just students, distinguished guests, it is indeed my pleasure to be here today for this occasion. I am glad and also appreciate in a special way this institution, because down here where we come from, people gather mostly for monetary businesses and political interests. People no longer have time to seek for knowledge that will help to save the country from the doom we are facing. I could remember when I went to search for a job in a polytechnic. The man said I should go back to school and read more. I said, I have the money. He said, money won't be a problem if you are telling me you do so. That in time, that people will understand life better. I will not take matters to court. They prefer to go to the church or the church ministers who will settle the issues, and there will be no imprisonment and no fine. And people who will be knowledgeable enough to take care of their income are not accountants. That the only people that can recreate nature are scientists. And because you're a scientist, that will like you to go back and seek for knowledge. So I am glad that in this part of the country, institutions don't believe in investing in knowledge. When you talk about seminars, conferences, schools only think of what will bring income. But I'm glad that this institution today, we need to applaud them for the courage, the support they have given these young people to present this symposium. Thank you very much, sir. I will begin also by commending these young people. A lot of us are looking for how to reach our pocket, but they are determined to give back to society. Most of us didn't have the ambience, but because they have chosen to give back to you a little bit of what they have observed, experienced in life for the future, I also need to commend them. But because of time, I think at this point I will go back to the issue here, which is leveraging RNA research towards career development and therapeutics. Sometimes people begin to wonder why this aspect. Before now, biochemistry was just seen as the chemistry of life, but we observe that life is controlled and determined by little molecules that make up the entire system. And when we look at these molecules, you see that each of them has a specific role. We look at the roles, we also see that in the components there are some that play in the elemental science, we say all the wears and tears, that play all the mechanical roles in the body that bring about and sustain life, proteins. Which means that what we have today is a manifestation of the proteins of the body. And these proteins have a machinery from where they are produced. And all the things that happen in life, proteins mediate all the life processes. Even as I'm talking now, your muscle, protein. Enzymes that convert fats to protein. Hormones, protein. So it becomes important that if these protein molecules actually determine the functional state of life, what happens to the system, which can lead to normal life, healthy growth, or disease conditions, it is possible drugs we have been using all along are what we call THERAPEUTICs. There are foreign bodies. The body sees them as a foreign body. The body also acts on them as a foreign body. In their breakdown, there are intermediates that also cause harm to the body, toxic. They also constitute burden for the liver, because the liver has the capacity to detoxify the body. So, but when you look at this, we now see that even drugs on their own are not healthy. These synthetic drugs we take are no longer healthy, which is why now we are looking at natural products that will not place burden on the system to ensure that when they are consumed, they not only affect or alleviate the condition, but they can also add value to life. Which is why we're now looking at new nutraceuticals. But beyond that, just observe that there are some conditions that we call terminal diseases, which we term undruggable diseases, like cancer. See that for such conditions, no drug has been known to be efficient. We have chemotherapy, surgery, and all those to see if we can reduce rate or remove them. But in terms of controlling them, since they have to do with cell proliferation, which means the way they divide is not regulated, and this regulation is related by proteins, then we can see how we can work on those proteins to see how we can use the machinery to regulate all these conditions. In which case, it becomes what we call personalized therapy, which means for each person, because each person has a unique genetic makeup. We look at the system, know what the problem is, then engineer the synthesis or inhibit the protein so that that condition will not prevail. So on this note, I present the outline of the talk today, and then we can just move. Next slide. Now, what we have today is what we call the African Life Science Salon. It's a network of young scientists who have discovered the novelty in RNAs to see how we can use to unravel the mysteries that can help us treat some diseases and conditions. So this is targeted at exploiting the potentials of nucleic acids research with special interest in RNA for therapeutic options. Like I've said, we have 4 major macromolecules: carbohydrates, proteins, lipids, and nucleic acids. Nucleic acids, they are of 2 types: DNA and RNA. And of course, you know that DNA is a repository where all the characters, all the threads in the system are stored in specific locus called genes. As well as defined genes as specific locus in DNA that code for specific characters. Now they are carried in the chromosome, but because they carry these codes, but this code must have to be translated into proteins that will then elicit those functions. Next. So now, you see, we are now delving into what we call the province of biochemistry and molecular biology, because all along it's been concepts and ideas, but because of the fact that we understand the mechanism of actions of things in the system, then that is why the American Association of Biochemistry and Molecular Biology described this area as unlocking life's secrets. Next slide, please. Move fast. Next slide. So now we have what we call early, and one of them that's prominent here is the genetic hypothesis, which says that everything we see that manifests in a system in biology, we call it a phenotype, is determined by what is contained in the gene. Which is why we are going into the gene, we're going into nucleic acid to see how we can use that to improve our life. Next slide. In this circumstance, I think we can talk freely if it's not forthcoming. Now, first of all, we need to look at what is the one that codes for all the characters in the system. Then the other one that is the RNA has a single strand. The difference is just that the DNA contains a deoxyribose sugar, while the RNA contains a ribose sugar. It's a 5-sugar molecule, and they make up what we call the nucleotides, which are composed of the sugar molecule, nitrogenous base, and a phosphate group. Now the arrangement of these molecules is determined by codes, and each of these codes we call codons determine each amino acid. That's the way they are coded determine it corresponds to a particular amino acid. And when we talk about protein as being the if you have a vehicle now, apart from the fuel, the entire wears and tears of the vehicle, the tire, the horn, the headlight, the seat, everything is protein in the body. And these proteins are macromolecules made up of amino acids with different features and characteristics. Now, the sequence of amino acid determines the nature of that protein. The hair is protein, the muscle is protein, the enzymes are proteins, but their differences are determined by the nature and the type of amino acid and the way they are arranged to form either primary, secondary, tertiary, or quaternary structure. We're not going there now. Now, if that be the case, we then know from our science that there is an organelle, a small organ in the cell, that has the responsibility of protein synthesis. I'll call them what? Ribosomes. Right? That is where they are important. Then this RNA are of three types. We have the ribosomal RNA, whose duty it is to form the proteins, the covalent bondings of the amino acids of protein. We have the transfer RNA, whose duty is to be charged with the required amino acid, depending on the code, to transfer the amino acid to the point of synthesis. And we have the messenger RNA. Now, if you now break it down, you now see the DNA has one property that is unique to it. It has the property of self-replication. And since it is a double strand, there is what we call complementarity. It means that one base pairs with the other in the chain. And we have 4 types of bases. We have adenine, we have thymine, we have cytosine, we have what? Guanine. Now the difference between RNA and DNA is that in RNA, you can say replace what? And that is the major difference. Now, adenine pairs with thymine with how many bonds? Double or triple bond? C to G is what? Triple bond. Okay, I just want to refresh your memory. Now you see, in that manner, you now see that we have what we call central dogma of biochemistry, which means that the DNA can self-replicate. They carry the codes, which is similar to what when people have commercial schools, we have what we call shorthand and longhand. The transcripts are what we call the shorthand, which is eventually translated to longhand. Now, if we now come back to that, I said the process is not of DNA self-replication, which means the DNA can unwind 2 ways. It is either conservative or semi-conservative, though there's a third one anyway. So, in doing so, the unwinding are made by enzymes, helicase and co. But the thing is that when they unwind, they expose what is contained in this strand, or what's contained on this strand. So that during the replication, then since we already know that A pairs with T, C pairs with G, the complement will be carried to form the 2 strands that will eventually become the DNA of choice. Now, continue before we've gone beyond this. Okay, now, apart from those 3 RNAs that the functions are already known, then it was observed that in considering nature of RNA, there are other types of RNA that have smaller fractions and whose functions, as it were not known. We never knew that they had what we could use them to achieve. We have one, the small nuclear RNA. It's not involved in protein synthesis, which means it's non-coding. But in RNA processing of cellular structure, which means it is involved in what we call post-transcriptional modifications. Then we have small interfering RNAs, which is one of those areas of interest. In this case, RNA can also form a duplex structure, and that duplex structure can enhance its use in this circumstance, because this small type of RNA has the property of silencing certain sequences. Before now, it was difficult because silencing was thought to be such that the entire function of the protein was silenced. But it has become obvious that with the help of this small interfering RNAs, that you can choose the particular process of protein to silence, making it become selective. Then, the fact that RNA can form a duplex structure, resembling the best pairing of RNAs to form that duplex structure. We also have non-coding RNAs. These ones, initially, the problem was that because of the fact they are long and bulky, it became difficult to incorporate them into the cell. But if you eventually we observed the nanoparticles and lipid layers that became available to be used to deliver them into the cell. Go ahead. Okay, continue. We already know the end. All right. You see, the first approach of RNA-based pairing therapeutics focus was that of an antisense oligonucleotide. We call them ACO, targeting some sequences in a virus. It was observed that these were able to inhibit certain pathways of viral replication, just like the HIV virus. The problem of HIV virus is that we already have the three major proteins: the integrase, the protease, and the reverse transcriptase. But it has been possible to get drugs that can inhibit the integration, that can also inhibit protein synthesis. But there's no drug to inhibit the ability of the virus to write back DNA from RNA with the help of the reverse transcriptase. Now, if you now look at this effect, you now see that biochemists have observed that these small structures, the formation of antisense and coding and non-coding ends, because in the structure of the DNA, we see that once they are produced, we have introns and extrons. The extrons are the ones that are coding, but the extrons are the non-coding because they occur at the end, while the coding sits at the middle. It was not as if they don't have any function, but it was later realized that they have function. So the essence is to see how we can use this understanding. Let me see if I can reach this place so I can not to eradicate or treat. And secondly, it can provide a pathway for intellectual development. Now you see, most of us have dwelt on, most students want to read law and medicine, believing that is where the world ends. But in the biochemistry, today we look at medical biochemistry, work in the hospital. We look at food biochemistry to just be in maybe next to. But people do not understand that the world is changing. Now, this concept came into being is because of the fact that COVID-19 became a pandemic that went to the world. Scientists went through the process of vaccine production and saw that one, it takes time. I'm not sure the same vaccine that was produced within 10 years because of the stages in the production, from discovery, lab trials, animal studies, human trials, clinical trials, and lots of them. It takes over 10 years. Then, drawing from stem cell therapy, where we can induce the cyst in the bone marrow to produce certain cells that can mediate certain conditions, it was observed that also doing that to proteins will solve most of the problems. And that was why it was possible that within a very short time that the vaccine for COVID-19 was produced. So I off my mind. But we can just select the gene. I mean the antigen of the microorganism that has that antigenic stimulation, then produce a replica of it or a protein of some same structure that can be injected in the system to induce antibody production that will work against that antigen, because in immunochemistry it's about antigen and antibody reaction. So when the antibody is produced, it will be able to react with the antigen in an observable way. Of course, there are three ways, but we won't go into that. So it became possible that we can induce protein synthesis in that direction. We also know that in cancer is about uncontrolled cell division. You see, the cells divide naturally, but there is an extent to which a cell will get to and it stops. One could be by cell-cell contact. There is contact inhibition. Another one could be by regulation, gene regulation, protein synthesis, which means that if a certain protein, if there is mutation and the wrong protein is produced, it is possible there may be issues and there will be uncontrolled cell division that constitute the cancer today. But if we can also induce certain protein synthesis that will either inhibit or promote certain pathways, then we can also check the activity of those proteins that will lead to the uncontrolled cell division. We have also observed that we've been told that there are some women in the world that no matter how they are exposed to HIV, will never have HIV. Right? You all know that if you look at the structure of HIV, the virus has completely shaped with the docking site of the cells. Now, what happens? In the cell, we have what we call CD4 sites, clusters of differentiation, where the virus latches with its glycoprotein 120, GP120. In that latching, that docking alone is not enough to cause an infection. But when that happens, there is a change in the configuration which will enable it to bring out another fusion peptide using the lysine side chain to bind to another site. Another site is close to CD4 that's called the co-receptor site. It is only when it binds on the cell, both binding is effective, that it can layer this portion and the virus will go to inject its content to now infect those cells. And CD4 cells, they express that docking site. That is why they are the cells that have been attacked. CD4 cells, macrophages, monocytes, and also them. Now, in that other point called are found in those places. Now, if you have a mutation that instead of having that CCR4, you have another kind of chemokine that binds, that is found in that site. It then means that it will not allow that second binding. And for us to understand the nature of the chemokine and induce mutation that will lead to the production of that other chemokine, then it can inhibit the second binding. And for as long as life exists, that virus will not be able to infect the site. Now, these are the issues that are being considered, rather than drugs that have side effects. So, in quick case, we find useful application of this RNA therapy. They are proteins. These proteins induce proteins. They induce synthesis of protein that will assist certain functions in the body. We also have in cancer treatment, like as I mentioned. We also have in agriculture, inhibiting and also allowing certain threats. We also have them in even forensics. That is why it is necessary that if we understand the gains and prospects of RNA therapy, we can find useful application for the future, so that we will not rely anymore on drugs. And as we all have our unique genetic composition, then what works for me will not work for you. We have in our chromosomes, we have how many pairs? 23. Then 22 are autosomal. That means also characters. One is sex linked, the one that carries the X and Y chromosome. That means maleness and femaleness. If you have such a condition, which is why it is sometimes seen that we have certain range of treatments for males and females because of those drugs may be relative to your body size, weight, and body composition. But when it comes to RNA, it is targeted to your own personal genome because already there is a genome project, courtesy of base research, that the human genome has been sequenced. That most cancers and drugs, they already know why it is possible for you to go to the chromosome and then know the DNA and know the locus, the gene that is responsible for any character. On this note, I encourage us to note that this is the science for the future. This is the science that will save the world from side effects of drugs. And when that happens, the side effect of drugs will be eliminated. The time is short. But the problem we have in this country is the political will to allow research to go on. In other countries of the world, they allocate so much money to education, 20% based on UNESCO standard. Nigeria has the least. And because of the nature of this country, our country is not driven by knowledge. Our country is driven by mafia. Because if it is driven by knowledge, we will have a good base for knowledge, a foundation for knowledge, in which case education will be well funded. There will be research and development. Right now, what are we using here? Things, factors that determine the expression of characters, which can be used to correct these conditions and even improve human health. So on this note, I want to encourage us to key into your society platforms. There are calls for collaboration, for scholarship, that if you develop an idea, people out there who understand the value of knowledge are waiting for it, which is why all of us are doing what? Jabbering to the foreign country. But for us, and as the only policy, the only tool to which we can save lives tomorrow, improve living. Thank you very much for being here.
That was a wonderful session. Ikechukwu Okorie. So you see the word. Prof, thank you, and we will keep having more insight into what we have and do. Get ready to mount the stage. Thank you. 5 minutes.
Our reasons for being in this place today. Father, we present this scholar to you. We cannot start eating it unless you sanctify it. Lord, we pray that you will bless this scholar before us so that we eat it and eat out life from it. Eat out love, eat out King of Glory, or more understanding from what we are doing today. In Jesus' mighty name we pray. Amen. Our Father.
We tell ourselves how we pick it up. So, Prof, the floor is open for you. Thank you.
I want to stand on the existing project and I'm highly glad to be here in this midst of academic giants to learn. The topic given to me, biology education, fostering tomorrow's RNA researchers through innovations in biology education. And when this topic came to me, I felt that it came at the appropriate time. So despite all the passion I have, I said to make out time, come and say one or two things about this topic. Also learn one or two things from other experts that are coming to delivers some lectures. At least today, I've learned enough from Professor Korea. So I'm very happy to hear from you. Thank you very much. And definitely the topic you have presented reveals some of my problems. But some of the things I have just said here are contained in what I will have presented. So I will just touch them in the passing so that our time will be economically used by us. First of all, let us look at who a researcher is. A researcher is someone that is an information officer. Not just information by hearing and saying, but the one that gathers information through his who displays information through gathering of information, through research work, to investigate the truth, analyze it, organize it in such a way that it will be useful, and then communicate to those who need the information, or those who can use the information to do something for the society. So in research, the ultimate aim is to solve problem or prevent problem. That is actually what we do when we are talking about research. So the researcher, makes his findings based on the investigations he has carried out, targeting a particular problem to be solved, or targeting how a problem will be prevented. So that any research we are carrying out that doesn't have these two issues at heart is not just a research work. So you must have a problem that bothers you want to solve, or that you perceive that is on the way that you want to prevent. And the overview of RNA is still contained in the what I've submitted, but then academic example. For those who eat oppa, I don't know whether you know what's called oppa. In early, but in recent times, they will tell that this oppa contains vegetable. That vegetable becomes what? Something different, added value to the oppa you have been eating. Because all days I was a child in primary and secondary school, my mother never cooked oppa that contained what? Vegetable. But these days, you cook oppa. You buy your oppa from the market, you see what? Vegetable. So we even add egg to it. So that is adding something new to create additional value. That's what is called innovation in the simplest language. So, in the field of academics, we think of the problems we have not been able to solve from what we have been doing. Every year, people enter to one particular level of academics, graduate. Next year, people will enter. Sometimes people can graduate. And we will continue perceiving, expressing one particular problem or the other, because we have not been innovative. So, in the field of academics, we now think of what are going to do new, so that we add added value to what we are teaching the children. And that is what we are talking about, what we mean when we say innovation in what? Biology education. What do we do in biology? What should we do in biology? From the cradle, about those for the future. Not to me again. I may not be able to go down to start learning something from what I should have done in secondary school, so as to make better for Nigeria. Maybe I can still do it, but for now, it's better for those who are what? Younger, who will be willing to, who have that enough time to undergo the process of research. So, innovation can be defined as a process by which a domain or a service or a product is renewed and brought up to date by applying new processes, introducing new techniques, and so on. So as a create what? Added what? Value. I just want to leave this and presence here because I'm not what I've said so far. So findings in science and technology need to be disseminated to those who will now enshrine into the curriculum, such that something new will begin to happen on what we are teaching for our future researchers. So innovation in biology education can be in form of new topics. We call it learning materials, that's for education. It can even be in form of new personnel teaching. It can be in form of new teaching methods. It can also be in the form of new instructional materials. There are many dimensions of education inside the education system. For instance, I'm a biology teacher, but I don't know how to, do one particular thing that can create employment for a child, and other people can also do it. I will say, okay, I'll get someone that's a local technician in that area. Maybe I don't know how to do fish farming, and my child is someone I'm teaching you to do what? Fish farming. It's a topic in biology. I can get a fish farmer and say, please teach those who are doing this thing, and they will learn it. I'm aware that where I teach a lecturer there, a case in the chemistry department, where the students need to learn how to produce gunpowder as undergraduates of chemistry education, and nobody knows it in the department. I told them, look for local gunpowder producers to come and teach them how to make gunpowder. Pay for that, and that's innovation, bringing in a new personnel into the system. So innovation can be in different dimensions. So that being the case, I want to leave this area and to move into other areas. Okay, we are saying that we should look at nucleic acid concepts and overview, but my lecturers have done justice to this. Hence, I have to give you here is a little, sorry, it's far below what he's given to us. He gave it to us in what? In details. And I don't intend to spend more time looking at the nucleic acids, how they differ from DNA. How we divide them into DNA and RNA, and then how the RNA differs from, have 3 types of RNA and so on. But from the work I've submitted, I was able to make differences between DNA and the RNA based on the structure, based on the chemical composition, and based on the functions. So they are contained in the material that I've sent on soft copy and which I have here, but these ones will not be necessary anymore. I'm aware again that even when you are teaching us how we classify RNAs, you touched the messenger, the transfer, and ribosomal RNA. I have all of them here, but I think we have done justice to that. And I'm aware that we also have what we call the coding and non-coding RNAs and so on. So that means what I think I used to know, what I wasn't sure I used to know, I'm now sure that I know them. Thank you very much. So we are going to look at how we can foster RNA researchers. How do we foster RNA researchers? So to foster is an English word, I mean, but it's not a scientific language. Never is it a research language, just an English word language. Foster means to nurture somebody, nurture something. If you are fostering criminality, that means you are nurturing what? Criminality. If you are fostering goodwill, that's you are nurturing what? Goodwill. So if you are fostering a researcher, it means you want to nurture a researcher. Somebody that will make out time to look into a way to solve problems in the society. So let us have a brief overview of fostering of RNA researchers. What we mean here is that what shall we do to have people who are interested in carrying out research such that all those benefits of RNA research, which our brother have just told us here, will be put into practice. That is the major thing we are looking at now. What shall we do to do that? Because Nigeria, as well as every other undeveloped country or underdeveloped country, needs people who are willing to go into research to solve solve our medical and agricultural problems. But these are the two areas we can apply RNA researches. So despite all the needs we have, because every innovation you are doing in society, you must consider the need of society. You can't just innovate when there is no need. So having innovation practice means that there is a need for something. That's what we're supposed to identify. What are those things that society needs? Once they're able to identify that society needs, that is the gap we want to fill. That is the vacuum we want to fill. Then you can now go to research to find out what you're supposed to do to fill that need. So, and then we find out that our teeming population, we are running shortage of food, and we are medically unfit globally. So that we need people that can think outside the box to find out what to do to stem that pressing need for food and that pressing need for medical, to take care of our medical challenges. So, and we start from the cradle, that's from the grassroots. You don't just come and pick a professor of biology education, for example, say let me train him on RNA. As I'm here now, I'm already getting out of work. This stage, highest 10-15 years, I will be resting in my village because the people was alive. But look for those who are going to be trained so that they can now start from the cradle and begin to give benefits to the society. So we also talk about those in higher education. At a point, I started looking at those who are going to speak today. I found out that something is missing. That we need to have invited some of the political elites, those who take decisions in government sector, final decision. Because after everything we do here, we raise up a community we are going to pass. But so far, there are those who are not political, who are not in the middle class, are not here with us. Whatever we say, they say, academic people, we don't went forward. Well, I feel that in subsequent gatherings, people in the political class should be invited so that they will understand what we are trying to say and they will have empathy with us. So that at the end of it, those undergraduates that want to carry out research, or those PhDs that want to carry out research, let's just make a budget for them. Because we don't just foster with empty hands. We need logistics, we need counseling, and we need everything to make a researcher to be trained so as to solve. Those who want to foster should be given logistics, as I said before. They should be given adequate curriculum materials, psychological supports, and there should be a form of monitoring and evaluation of the learning process. You want the learning process, not just say you are doing them to carry out research, you overlook what they are doing. The type of thing most of us do in the university system. Approve a top student to go and carry out a research, and we will wait for them to come and give you what he has written, whether genuine or not. Okay, the English is okay, idea is logical, you approve. Did the person actually go to the field to carry out research? We don't know, because we don't have adequate monitoring and evaluation techniques. So this time around, we are supposed to have such a situation whereby you give someone a research grant. You should be able to monitor what that person is doing. So before then, after then we look at those who want to nurture as researchers. What qualities we need to have from them? What are the qualities of those people we need to say these are our future researchers? What are their qualities? I have to adopt a work by Thomas in 2023, where the quality of a good researcher was actually discussed. But I have to summarize them into 10. There are many of them, but I have to summarize them into what? 10. And I'll discuss them briefly, but that's actually part of the course of the matter we're having here. One of them is that anybody want to nurture as a researcher should have passion for research. That should have what? Passion for for research. Not just that I want to carry out a course, I want to do it and graduate. I want to do it and take the money and be educated for me. You don't just say, because even in the university where I teach today, you see people applying for research grants, 1.3 million, 3.5 million, and say, thank God. Once you give you 1.5 million, 3.5 million, you got first of all repair his what? Car, buy new tires, repair the house, and then after 3 months, you give you a document. I have finished the research work, and then find that nothing can be done with that he has submitted. So that does not what we are looking for. We are looking for those who are interested in coming up with something new, something that they can swear that I have gone to the field, this is what I discover. Implement it and hold me responsible. That is the passion I'm talking about. So there should be a way we should identify those who have that type of passion. So in science, to be precise, passion and research are two sides of the same coin. You can't separate the two. That somebody is driving a car today is because somebody had a passion of finding out what you can do so that a car will be in existence. That you are right having your handset today. So somebody had a passion of what can I do so that I'll have a handset. So that should handset in existence. So passion and research are two sides. Curiosity. For you to be a researcher, you have to be curious. Curiosity means there should be a way you should be questioning everything you see. You question it. Why did this thing happen? Remember that if you look at the history of Isaac Newton noticed that each time an apple will fall from the tree, boom, who will pick it? Boom, who will pick it? And everybody noticed it, that apple falls from what? The tree, and people pick them. And nobody cared to know why the apple will fall from the tree. Why not when the apple detaches itself from the tree, it will start going up? Why not? Why must it start going down? So among the millions of people that saw that apple detaches from the tree and falls down instead of going up, only Newton cared to ask why. Why not go up? Why must it always go down? And from there, he now thought about his what? Force of gravity, law of motion, and so on. So that should that curiosity for you to know, for you to find out why certain events are like that. Even in social sciences, why is it that after every whatever exam is about to start, you know, there will be violence. Whenever it's about to take start, there will be what? Violence. Does that raise violence? You have to ask yourself what? Why? Okay, so then let's talk about perseverance. Perseverance means that research is time-taking and also painful to have to take the pains, and then make sure that you don't give up somewhere along the way. Some can carry out a research work, and before they can finish, they will meet frustrations and they will drop it. You don't need to drop everything you see. Drop the research work you are carrying each time you encounter what? Problem. Talk about also critical thinking. The researcher should be what? Skillful in analyzing data, identifying some patterns, and then drawing some meaningful conclusions. So you should be able to criticize your work as you are going on. So that whatever what you have in mind that you are going to put forward, that you have in mind, this is what you are seeing. You should be able to compare the two. The researcher should also collaborate with people or even a team of experts in that area, trying to find out the answer to a particular problem. Then next one is effective communication. You should be able to that thing you have found out. When I define a researcher, I say research is what? An information. That even the novice will understand you. Even what? The novice will understand. In those days, one of my lecturers told me that if you cannot explain a concept to an illiterate, that means you have to understand that concept. bring it as a teacher. As what? A teacher. If you can't explain any concept to what? A stagnant illiterate, it means you haven't understood that concept. Out of the people I encountered as a couple, when I was teaching about genes, SST private examination, they asked me a million questions about genes. You know, it's an abstract concept. All they say is it blood? I say it's not blood. You know, it's just I have a story for another day. It was a problem as a couple there. What is gene? After explaining this, they said, "But why do you say that if somebody is a thief and son is a thief, that is from the blood?" I say it's not from the blood. It's from what? The gene. Let's leave that matter for now. There's no time. So then you should have organizational skills, being able to organize whatever you are discovering in such a way that it will make sense, not just publishing or dispensing information. You know, it must be sequential. Then there should be prudence in whatever you are doing. Prudence means that you should exercise a very good judgment and caution in managing the resources you are carrying out for the research. Then next one is self-reliance. It's another English word. As a researcher, you have to trust yourself. Self-reliance does not cancel collaboration, but at least if you are working as a team and a particular session is allocated to you, you should be able to be confident to carry out that area that are allotted to you and be able to defend it. So you should believe in yourself. Someone who doesn't believe in himself can never go into research. Then finally, ethical conduct. This is where many of us are lacking. I just mentioned that someone can say, let me collect 3 million naira grant. That grant you got that research that gave you 3 million naira grant. Have you carried out this research wisely, judiciously, or even religiously? Do you see yourself as someone that has justified one that was given to you, or have you taken your own share of national cake? And then that thing you want to publish, is it real or is it doctored? Do you doctor the report based on what you feel is supposed to be, or do you bring the report based on what you saw in the field? So this ethical conduct is what we should take serious. So in conclusion, and aware of linking this to my own field, biology education, we are saying that the needed attention towards RNA research should begin by creation of awareness about RNA among secondary school students. That is where the thing should start from. At least I've got to the secondary school and I've seen the curriculum. Are you following? And I noticed that very little is mentioned about nucleic acids in general. Very little is mentioned about nucleic acids in Nigerian secondary school biology curriculum. And then the worst is that nothing was mentioned about RNA in the curriculum. You can pick it now, you can go and verify. But good textbooks contain RNA, DNA being in textbooks. But look at the curriculum used in teaching, which just now used to implement the curriculum, the teaching process. You don't see them there. And that means students that are supposed to learn this in higher education, we meet them for the first time in the higher institution level. So there is no background information that they have. So that is why I thought that if I'm going to have a company here for this symposium, something of that should come up. That it should be included in the Nigerian secondary school biology curriculum, at least fundamentals or elements of nucleic acid studies, including the RNA and the DNA. Then that will now give them the pedestal upon which they can stand and master RNA studies at tertiary education level. Then, in focusing the researcher for the future, our educational system should put into consideration all those mentioned qualities, because as the qualities I've just mentioned are better implemented at higher education level. Curiosity, prudence, all those things, they are better now nurtured or put those students during higher education level. We should have biotechnology, applied biology, zoology, plant science, and so on. They should, when they are talking about research, these qualities should be in them, so that the syndrome of copy and paste, which most of them do today, will be phased out. So, and then the Nigerian power brokers, you mentioned, there's a group you mentioned, you say cabals, Abi? The cabals, we should have a way of breaking into the cabals such that the researches of this nature that concern the RNA and other nucleic acids should be made manifest in the school system. I know that people are talking about RNA. DNA has been very popular, maybe because it is being used to test, look at the paternity, DNA. But RNA, which seems to be more sensitive, is not being talked about at all. So with our curricular innovations in biology education, this problem could be solved. Thank you very much for listening.
Prof, I said many things. He has it all. He's a diverse scholar. So please, a woman who is going to call women to stand up for science. I'm talking about no other person than Dr. Mrs. Antonia Nkiro Mba. She's the HOD Microbiology and Parasitology, David Umahi Federal University. She's a seasoned scholar. She's a representative of the woman in science that you may be looking for in overseas. We have it here. She has performed excellently in research, and you will hear her speak today. So that she will be a courage to other women who think that teaching can prevent them from achieving their dreams. Just listen. At the end of the day, during your feedback, you will have something to tell us about the new choice you've made about your career, particularly the female gender. Thank you. The podium is open for you, Doctor.
Good afternoon, everyone. We're starting on the existing protocol. I would like to start by thanking everybody, and particularly Mr. Alko. This is my first time of joining this program. So that means I'm just meeting every other person today. And I thank you all for the reception. And I'm glad to be here. Well, I may not really spend much time, because I don't really have much. I'm just here to call on women to stand up and let us take our place in research. Let us try as much as we can to not to let the men take the whole space. At least let us be well represented. We're represented, but not well represented. So let us stand and take the space that we need in research and in science. So I'm going to speak on this next slide. I'm going to speak on women calling women to stand up for science, inclusivity in RNA research. Before I say anything about RNA research, I think there is need for me to define inclusivity. What is inclusivity? Please, next slide. Okay. Let me use my phone. What is inclusivity? The dictionary definition of inclusivity says it is ensuring equal access to resources as well as opportunities to everyone, especially those who are at risk of getting marginalized. Equal access, opportunities. Equal access, that means whatever you are given as the male researcher, I as a woman, a female researcher, should have the same thing. There should be no gender inequality. I should be given policy of non-elimination of participants, ETC. That means when there is an opportunity, any opportunity, research opportunity at all, it should be open, to be made open, should be shown open. Shouldn't be a situation where they would say, you are a nursing mother. As a nursing mother, we will not take you. Or there should not be a situation where the female will fill a particular form while the male will fill another form. Then in the process, they are trying to, give the male researchers more opportunity than the female researchers. No. Inclusivity says everybody should be given the same equal opportunity and everything should be made open. And when I'm looking for grants, I'll be asked to write how many children I have and their ages. By the time I give the information and they find out I still have little babies, they will have disqualified. Then the male will go. And it's not as if the male, they do more work than the female. Or do they do more work than us? No. They don't. Because when they come back, they watch television. But when we come back from work, we go into the kitchen, we do the house chores, we take care of the house. We still come back to take care of them. Can't you see where we should be? That shows we should be given equal opportunity to, showcase what we have. So that is all. That's what I have about inclusivity. With this, we all know what inclusivity means. Really visible. Sometimes you see women who make some achievement in research and science, and they are not being acknowledged. An example is before I go into that, another studies or another information also showed that women in science are just about 28% of the total percent. That means men have taken up how many percent? How many percent? 72. What are we doing? Is it fair? It's just unfair. It's just unfair. And we need to do something about it. This research and part of science. This gender inequality or this opportunity research. There's this notorious example about Louis Chow and Sarah Levi, two scientists who did the first work on RNA splicing. And it was very unfortunate. During the 1993 Nobel Prize giving, they were not recognized. Even during the 1997 and 2017 anniversary celebration, these people were not still recognized. What does that show? Women are being recognized, and this calls for support. Calls for support for the women, because the women are always under the men. And even if we fight from now till tomorrow and they don't want to stand up for us, we're fighting in vain. So we're calling them to help us. Then, apart from all these, despite all these challenges, we have women who made advances, who made giant strides in RNA research. One of them is Dr. Katalin Kariko. She is a biochemist, and she pioneered research in messenger RNA research. She helped in the production of a vaccine against COVID-19 during the 2019 pandemic. And by doing so, she was able to save the world from COVID-19 threat. And with this discovery, she gained an honorary doctorate degree. And through this, other researchers are now exploiting the use of modified RNA in treating diseases, just like Prof told us. So researchers now use RNA, modify them what she actually did was to form a group known as UCC Future Medicine in University College Cork in Ireland. And this woman, with what she did was to get a team of world renowned leaders, leading scientists, engineers, and clinicians. That's we brought people from different areas and formed a team. And with this, they are actually working to produce transformational next generation medicine and medical technology. Next slide, please. Then advances. The first researcher, what's her name? Dr. Kathleen Carrico. The second one, Professor Catrino. Both of them are lecturers in UCC, that's University College Cork in Ireland. With their studies, they have been able to, carry out research to find out how to detect diseases earlier than when we were using ordinary drugs, conventional drugs. They were also able to, find out faster prognosis and targeted treatment for reduced and global health burden. And with their research in RNA, improve quality of life for patients with chronic diseases such as cancer, cardiovascular, gastrointestinal, immunological, and inflammatory diseases. Next slide. Then we also have another lady, still in the same school. Her name is Dr. Martina. She is another vibrant lady in the same university. She is a member of a particular group who came together to also carry out research. And the research they are doing is focused on how RNA is decoded into protein and how the process is regulated. All these things they are doing is really actually working towards helping in treating diseases and giving humans better life. Next slide. So then in the same school, a rapidly growing corporate startup company known as Bio provides a comprehensive analysis of messenger RNA translation at genome-wide level to pharmaceutical companies and drug discovery companies. So people who want to set up pharmaceutical companies or drug companies, they meet them and they guide them and give them foundation on what they need. So when you look at the discoveries made in this particular university by ladies, you will see that the ladies are not left behind in that particular university, even though ladies are having challenges. So what am I trying to say? Do we now sit down and fold our arms and say, because we're not being recognized, because we're not being given the opportunity, let us sleep? No. Next slide. So, okay, we still have other areas of research where women played central role in RNA research. Another one of them is experimental work on transcriptome changes in myotonic dystrophy type 1. This is a kind of a muscular disease that is being treated using transcriptome changes. Another discovery is the role of there's another work on RNA is the role of extracellular vesicles, that microRNA and transfer RNA in synovial fibroblastocysts. I think when Prof was during his presentation, he mentioned these types of messenger RNA. These types of RNA, that's the micro and the transfer RNA. These RNA can be, used in treatment of different diseases. Then, initially, some decades ago, we were taught that RNA is seen only in the nucleus and cytoplasm. What does it do in the nucleus? In the nucleus, it is synthesized from the DNA. Then when it's synthesized from the DNA, it is processed. The first, okay, it is synthesized into precursor RNA, then it goes through splicing and then processing and then comes out into the cytoplasm. So we were told decades ago that you see the RNA just in the nucleus and then in the cytoplasm of a cell. But studies in science have shown us, studies on RNA have shown us that there are also extracellular vesicles. When we say extracellular, that means cells that are outside the cell. Sorry, vesicles that are outside the cell. So there are also vesicles that are outside the cell, which means they are not in the nucleus, they are not in the cytoplasm, they are outside the cell, but they also contain what? RNA. And the studies in RNA research have shown that these extracellular vesicles contain RNA molecules, which can also be used to treat diseases. Then there was also another study on RNA, which showed the effect of Moringa oleifera microRNA on lipid metabolism, and also on the other researches. I'm just trying to show you that although women were having or are having challenges, that didn't deter them from doing what? From carrying on. They are still working, still trying to see what they can offer, whether they are recognized or not. Next slide. So what are the challenges? Challenges faced by women in science and research. Before I go into challenges faced by just women, there are challenges that are faced by everybody, not just a female, but everyone as a researcher. And what are those challenges? First of all, being a researcher is very challenging, irrespective of what the gender. Academics are facing increasing pressure to publish. There is this saying that it is either you publish or you do what? You perish. That means for you to grow academically, you must publish. Then if you decide not to publish, you remain there. So there is pressure for every researcher, whether you are a female or a male, to do what? To publish. Then, one, we're talking about publishing. Two, there is need, the rate at which you are required to publish now is on the high side. It's not just publishing. You have to publish frequently. And then it's not just publishing frequently. Like now, where I am, for you to be promoted, you publish in recognized journals. Not be like now we have what we call the bills list of journals, where for you to publish a good paper that has to be recognized, you need to spend money. There are journals they call Imago, Toxiruta, and the rest of them. Let me share one of my experiences recently. A few weeks ago, I came back. I had this paper I wanted to publish. So I decided to, check PubMed to see if I could publish one of the papers there. I clicked on one. By the time I opened and checked the price, you know what I saw? 800 and something thousand. I screamed. I said, I beg, this one is not for me. I just closed it. So there is pressure on publishing, and not just publishing, the frequency, and then where you publish. So you don't just publish and bring out anything. Gone are the days where you just go to any journal and publish it and bring it out. It is paper. It's no longer like that. Then another challenge is managing a scarcity of funds. How much do they pay lecturers? The people in the house, how much do they pay them? The lecturers, we work, and most times they say our reward is in heaven. May God help us. With our mega salary, you can see what we're passing through. Not talking about the gender this time, whether you are a male or a female researcher, we are still faced with what? Scarcity of funding. Then sudden reduction in grant success rate. Initially, people don't really know about grants. So anybody that goes for grant gets it. But today, everybody's eyes is wide open. Everybody knows about grants. Everybody's writing, and then the rate at which people get grant has really reduced. So that is also a challenge. Then in terms of those students who has a high standard, you know you have to give in your best, squeeze yourself out in order to impress such a student. And then on the other side, there are students who need your attention. You know, there are some students you need to nurture, need to guide. Because when you leave your home and come to school, we are your parents. We are the parents for you until you leave the school and do what? And go home. This is also a burden on us, which we have to do because we should be there as the parents of the students. So it's also a problem and it's also a challenge. Then opportunities like being the head of department, being the dean and the rest of them, they also sap you, squeeze your time. By the time you do the work and then come back, you get so exhausted that you may not really have time to, you know, publish. Then another one is study. Okay, study shows that the competition we have in the higher institution today is not as much as it used to be. Today we have a lot of competition. Everybody wants to be the best. Everybody's eyes have opened. You know, like I said earlier, you know, going for grants and the rest of them. So we notice that there is a lot of challenges and a lot of competition in the university and in the research system. Then these ones are the challenges that are faced by both men and women, male and female researchers. Then what of the female researchers? There are challenges that are peculiar to them, like sexual harassment. Then women experience higher work and family crisis or conflicts. You go to work, sometimes you have problem at workplace. You come back home, you face the one at home. And then you see it's very challenging, and sometimes it could be discouraging that if you don't take time, you may fall out on the way. But then, as a strong woman, what do? You tie your belt and sit up. Then we also have bias, both conscious and unconscious bias. Sometimes when they want to give out a particular work, they will say, she's a woman, she can't do it. Let's give it to a male. Or sometimes they look at you when they want to give out a particular post. They will say, this one cannot do it. They will look for a male. This one cannot do it. They will look for a male. Then, or sometimes even when you are given that job or that position, the male colleague in the department or in the school or your co-researcher who is a male may not give you the opportunity to express what you have. Then we also have an issue where sometimes when the male and the female come for a particular position, they give the male the opportunity, feeling that the male will do it better when it's not actually good. So other challenges, lack of mentorship and the rest of them. So how do we surmount these challenges? How do we surmount them? Are they surmountable or are they insurmountable? They are surmountable, yes. They are surmountable challenges. So, but then it starts with the major solution, or one of the major solutions is inclusivity. Because inclusivity encourages you to be there because it's the male. But when we come together, when everybody is given equal opportunity, we come together to do ideas together and we sell together. Then another way is to look for peer rhetoric and work together. Then another way of solving this problem is to ask for help. Don't be a loner. When you are doing a research and you are having problems, what do? Ask questions, meet people. Don't keep it to yourself. Nobody woke up one day and became the best. We grow, we learn by practicing. And when you do that, when you ask questions, you get ideas, your problems can be solved. Then another one is be confident. For some of us, when we want to do something, we may feel, I don't do it well, people will laugh at me. Please, drop that. Be confident. When you ask questions or when you take the step and you don't succeed, when you repeat it, the next time you see yourself doing better. But when you hide inside your show, what happens? You may not get it right. Then the last but not the least is collaborate with other researchers. Like I said earlier, don't be a loner. Work with others. Look for people you can collaborate with. When you have ideas, don't hide and say, let me do it alone so that I will have the glory alone. No! Collaborative work is what is being encouraged these days. So you collaborate with others so that you can pull ideas together. And when we do this, I believe that as female researchers, that the sky will be our starting point. Thank you.
Talking about a study on Moringa oleifera, I got interested because one of my first publications in molecular docking was actually that, and it has gained a lot of citations. And we are just engaging on how to discover nutraceutical targets for RNA diseases, and some of our studies are ongoing. So there is prospect. So can we give a round of applause for Dr. Mrs. Thank you. There was a technique. Thank you. Welcome to the podium.
Please, I'm not a great teacher like my professors here, but I'll try to do my best. Can we hear me? Sorry? Yes, I'm a teacher. I do more of what she said. Thank you very much for your presentation. You know, she mentioned a lot of gaps that we have with women scientists in Africa. Interestingly, they are doing very well abroad. In fact, when I was doing my PhD in France, my bosses were all women. There were 4 of them and they were all women, and I did very well during my PhD. So it means that women have a place in science, really. But African women have to take their place. It's not by sentiment, you have to work for it. So on that note, I think you actually need for mentors who will guide you through. It's not about publishing your work in good journals. If you want to get good mentors, get grants, then you need to do your ground job very well. If you're applying for grant, for example, now, you just get it because you're a woman. But if you have some good scientific output like conference papers, quality journals, etc., now the therapeutic journey of diseases, it starts from, okay, there are 3 major areas in disease control. You have, of course, the most important is prevention. Then, if it doesn't work at that level, then you have to go to treatment. But for you to have effective treatment of any particular disease, then you should have effective diagnosis. Regrettably, this is like the most or the weakest link in the therapeutic journey of diseases. It shows that in most hospitals, only about 1% of them have diagnostic centers that can give you what you want. And of course, if for any reason you don't get to know what is causing your problem, you'll be maybe beating about the bush trying to get the control or treatment. Is that right? Therefore, it means that diagnosis plays a central role in getting things right in the therapeutic journey. Now we look at the role of RNA in righting this wrong that we've seen so far. Okay, okay, that's fine, it's my outline. But the next slide, please. This means you get maybe blood samples, maybe plasma or serum, and look for antibodies or antigens that will enable you to say, okay, because I have these antigens, it means that I have a particular infection in this patient. That's serology. It can also be through microscopy. In this context, it means that you're going to get the patient sample, then make a smear. Look at this particular smear under the microscope. Most times, for you to get a good diagnosis using microscopy, and most times, there's this bias that you see with microscopy. Maybe if Mr. A reads the slide, you may see 2 parasites. Mr. B, you see none. Mr. C, you see. You can also have the
Yeah. You can also have the molecular approach where you talk about the RNA, the DNA, etc. Of course, it seems to be the game changer, but it also has its flaws. Most times, need to be very technical before you be able to apply this technique in diagnosis of diseases. Then we also have the other approach, which has to do with therapy test, therapy kits. We are all in Africa. Most times you have malaria, you just go to the hospital, they tell you, okay, let's just quickly check your NP level using the kits. Once it's positive, they give you anti-malaria. But most times, you may have issues with getting either a false negative or false positive result with particular kits. Therefore, it means that it also has its own drawbacks. I've mentioned about 4 approaches in diagnosis: microscopy, serology, rapid test, and the molecular approach. There are also other ones like the imaging. But in all, these different approaches have their shortfalls. Therefore, most times you need to complement one with another to be able to come up with a robust or convincing result in diagnosis. But of all these approaches, the most convincing one is the molecular approach. Interestingly, we're talking about the RNA Salon today. So we look at the place of RNA in effective diagnosis of diseases. Go down. Keep going down. Next slide. Okay, stop here, please. Now let's look at the diagnostic gaps. I've mentioned so many approaches, about 5 of them in the previous slide. If you look at this, means that we have some current gaps that need to be filled. Number 1 here is this issue of you won't be able to identify different strains. The microbiologists know that we have different strains. So if you're trying to treat a particular disease with a drug that's only effective in a particular strain, you may come up having failure because you're not treating the right thing. It means that you need to use a tool that will be able to identify the particular strain and species of that organism. By so doing it now, your treatment approach will be effective. Number 2 is also the issue of not being sensitive enough to tell you that it's actually the way it is. For example, some tools are like maybe like the rapid kits. Some of them may have been stored for so many months, and because of that, they fail in detecting these conditions. Therefore, may end up having false negative or false positive result. Therefore, they are not too sensitive. We also have the issue of personnel errors, like I mentioned before. Maybe 4 or 5 different experts giving you different results. Then lastly, we also have the issue of false negative or false positive result. And if you don't get these things right, no matter how well you try, you won't be able to get effective disease control. Therefore, there is need for us to go molecular to correct or to right these wrongs that we see so far. Now, we are in the tropical region of the world where we are ravaged by infection. Yellow fever, COVID-19, etc. We do a swab of our skins. Tell us what we see now, microbiologists. So many microbes, right? So we are at risk. It therefore means that for us to actually be on top of our game in control, we should be able to get diagnosis properly. I'll tell you a little experience I've had in my research surgeon. When I was in Gambia, just in February this year, samples were sent to our lab in Medical Research Council in Gambia from Cameroon. Some were confirmed positive for malaria. But when we looked at these same confirmed samples for malaria, we found something different with the molecular approach. It means that we need to actually use effective tools for us to get effective diagnosis that will also enable us to tackle these things head on. In other words, what we have talked about before now have their shortfall there for this. Need for us to go the RNA way. And I'll tell you why we should go the RNA way. Next slide, please. Okay, yeah, look at this place. Prof mentioned the central dogma in biochemistry. I won't dwell much on this because you already know what he talked about. Dogma. Okay, from DNA, RNA to proteins. Not all the DNA get to RNA. Not all proteins also get to. Not all RNA also get to proteins. Some of them are lost. Okay, but in this RNA, even though those that are not involved in coding, they call them the non-coding RNAs. They also play a lot of regulatory roles in what happens at the disease level. So through molecular approach, you'll be able to detect both the coding and the non-coding regions that will give you enough information about the disease condition. That's why it's important to look beyond just the proteins that you see that are maybe seen as enzyme, etc. Because some enzyme may not give you the real information, but if you go the RNA way, you'll be able to get to the root of that particular condition. Now you may be wondering now, why not use the DNA? Go to the next slide, please. Just click once. Okay, wait. Go back. I mentioned before that we have the coded and the non-coded RNA regions, right? So if you look at this, the non-coding regions like the microRNAs, etc., control about 60% of proteins expression. It therefore means that since most enzymes are proteins, if you want to know what really happens with these enzymes, you should also focus on these microRNAs. And you can only do so through RNA research. Next slide. Okay, just wait a minute. I was asking, I said, many people wonder, why don't you use the DNA as a tool for diagnosis? We all know this, that the DNA dwells mainly in the nucleus, right? But the RNA is more versatile. It can be found in the nucleus, cytoplasm, and also have the ones that are extracellular. It means that the RNAs are more versatile and can easily be found. Wherever they are, whether DNA, cytoplasm, etc., they can be used in diagnosis. Also, the DNA is more or less static, just confined in a particular place, but the RNA is more mobile. I'm going to compare the two of them. This comparison here now. For abundance, the RNA is more abundant. Therefore, we need a more abundant tool for diagnosis. When I was doing my research, I used RNA to confirm my results with proteins. What I did was to get the RNA through the RNA extraction techniques because they were more abundant in the parasite. Then did what we call reverse transcription, the RT-PCR technique, to now get a complementary strand to form the double-stranded DNA from where I now got my result. But my point here is that the RNAs are more abundant than DNA, therefore more useful in diagnosis than the DNA. Is that clear? Okay, next thing here is mobility. I think I mentioned this before too. They are more mobile, they move around freely in the cell. And lastly, because of the abundance of the RNA, you need fewer number of reactions to be able to get something quantifiable in PCR, unlike the DNA. But I have to say this very importantly. If you compare RNA and the DNA in terms of stability, the RNAs are very, very labile. They are very unstable. So when you're working with RNA in your research work, you need to work at almost ice level, like about 4 degrees. But for DNA, they are more stable. So just press once. Just one. It is stop. Then if you compare the RNAs with proteins, yes, the proteins are also very abundant, also very mobile. But the issue now is that not all the RNAs get to the proteins. Now different kinds of RNA, we have the messenger RNA, which are the ones that actually get to the level of proteins, right? So it means that Next slide, please. Okay, just a brief of the procedure for RNA diagnosis. I won't very much because I know you may be a lot of our time here now. Now, in doing RNA research, there are basically 3 procedures. The first thing here is to isolate the RNA from a cell. It could be from an ordinary procedure. If you make any mistake trying to work at room temperature, you end up getting a degraded RNA molecule, and that will not give you what you want. So you have to be very careful in your isolation process. Then for the DNA, cDNA means complementary DNA synthesis. RT PCR means reverse transcriptase quantitative PCR. We are looking at, let's go back to the previous slide, let's go back to DNA. The forward one is called transcription, while the backward one is called what? Reverse transcription. So in RT-PCR, you are doing the reverse transcription of your RNA transcript back to DNA. Okay? Okay, stop. No, go back. Okay, stop. Now, so once you are able to reverse transcribe your RNA to get a complementary DNA strand, from there down you perform your PCR. PCR means what? Polymerase chain reaction. That will enable you get more transcript of that cDNA. From there, you'll be able to quantify the level of the particular RNA there. Now, if you're wondering how would this actually help you diagnose or detect diseases? Okay, this particular RNA system that can help you to do 3 basic things here now. You can enable you to identify the aberrantly spliced genes. Number 2, it can help you detect allelic specific expressions, and lastly, to identify gene expression. Let me explain them one after the other. In the first case, she mentioned that before RNA become RNA or fully functional, they have to be spliced when you remove the non-coding part, right? Good. That's splicing. So when you do this RT-PCR, it can enable you to determine the wrongly spliced RNAs. And once you don't splice the RNA properly now, it can cause these conditions. Okay? Second one here is to detect allelic specific expression. Now let me explain this. Let me use the malaria in this case now. Malaria is caused by the parasite called malaria parasite or Plasmodium. Sometimes over the years of taking anti-malaria drugs, this parasite can acquire some traits that will make them different from the original ones. So with this technique now, those variations you see in the newer parasite now, they are called the allelic forms of the parasite. So you'll be able to find out those allelic forms of the parasite using this particular technique, and that can enable you determine whether there's issue of anti-malaria resistance in a particular environment. For example, if I come to, let's say, Southeast now, like we're doing at the moment, and I get samples from patients that have malaria infection, I look at the parasite strains, find out that some of the malaria parasites, they have a different makeup from the others, meaning that they have some variations that could be linked to failure in anti-malaria treatment. Therefore, it can help us do surveillance for anti-malaria resistance in a particular environment. Then, lastly but not the least, identify gene expression outliers. What this means that, it means that normally the gene should be expressed, move from the DNA to the RNA proteins, right? But sometimes, in the course of this expression, there could be some issues that does not conform with the norm. That's what we call the outlier. So with this particular technique, you'll be able to determine whether what you're seeing now is in conformity or an outlier in the normal thing. The next. Okay. Now, how can we apply RNA in diagnosis and also surveillance? If you look at this slide, you see that it can be used, one, to monitor or to survey for antimicrobial resistance. As I mentioned before, if you look at the genome of the parasite and find out that there is some aberrations in their genome, this can actually be traced to see if these aberrations are responsible for failures to anti-malaria. I'll give you an example. For malaria parasite, that's my area anyway. That's why I'm talking about malaria most times. If you look at the gene for the parasite, there is a mutation that occurs in the K13 region. K13 region. Once you have a mutation in that K13 region now, it makes the parasite become resistant to normal artemether lumefantrine that you already take every day, and that can cause maybe like I would like to call it failure to anti-malaria in that particular environment. Therefore, using this skill or two rather can enable you determine anti-malaria resistance in a particular environment. Okay, it can also be used in diagnosis. In this context, you are looking at just detecting a particular transcript in that individual. For example, in the case of COVID-19, I did PCR for COVID-19. They have to insert a swab stick into your nose or your mouth, get the sample. If they find the RNA transcript of that particular patient or that particular organism in you, it means that you are actually infected. Therefore, they can now commence treatment for that particular disease. Then lastly here is treatment. Just going by the same COVID again now, they also use RNA treatment to cure COVID. So which means that RNA research goes across the entire therapeutic journey of disease, from diagnosis, surveillance to also treatment. Just response. Now, if you look at this, the whole of this is called therapeutics. That is treatment and also diagnosis of diseases. So RNA comes across the entire length and breadth of disease management. It shows that it's very important for us to explore and exploit this tool in our disease diagnosis. Okay, in conclusion, we see that RNA is capable of changing the dynamics in disease diagnosis. Therefore, we need to adopt this particular skill and tool to enhance treatment outcome for our patients. Number two, we can also detect hidden diseases accurately. Like sometimes you may go for COVID-19, if you just use ordinary test, you may get a false negative or positive result. But with RNA diagnosis, you'll be able to get an accurate result. Thirdly, also have the issue of you can write the wrongs, like I mentioned before, because the margin of error now is limited with RNA in diagnosis. It means that all the wrong that we have seen so far now in diagnosis can be righted through RNA technology. And lastly, it can improve disease control and therapeutic outcomes. At this point, I think it's wise for us to think through what I have said so far. Maybe your next line of research should be thinking, how can I actually incorporate RNA into my research so that I will also contribute in righting the wrongs in diagnosis. Thank you very much for your time.
That was a very interesting session by Dr. Chinedu Ogonaya Ebu, an international scholar, MRSC scholar. If you want to talk about non-communicable disease, he is heading towards an expert because I know very soon he will become a young professor. So can we put a round of applause for him? So we will quickly take our lunch. After our lunch, then we have, instead of group discussion, we have a general question and answer session so that you can ask question and yes, tell the preacher participants to also send their questions. Our guests, we should also expect from those people are connecting, and I'm happy it's very clear. They are also sending testimonies from other parts of the world. They are with us. We have technical issues, but we were able to surmount it, and the future is very bright. So thank you. We are also going to share feedback form. We are going to share soft copy, and we are also going to share manual. When I give you just comment, actually the brainchild, we have come to stay. So enjoy your meal for the next minutes. So Prof want to say something.
Yeah, thank you very much. Please, it's not as if I want to. I love women. No, I love women. No, I don't know what's your mind, and I'll tell you why. You see, please. I want to support Madam because I was inspired by what she said. You see, while in the class, there is no discussion in teaching and exams about man and woman. I would just want us to keep busy with it until the food comes, right? You see, if you come to embryology, you'll observe that woman was actually created to be stronger than man by origin. If you now come to the molecular basis, if you take a survey of the X and Y chromosomes, their ability, capacitation, movement, endurance, which is the basis for sex determination, you will see that the X chromosome is stronger than the Y chromosome. If you also go take it further to the newborn, you see, one of the things that is used to assess strength is what we call neck check. When you carry a baby and the baby is able to raise the head, it drops. It's a mark of strength. I've heard that before. It's called neck check. If you have two children, male and female, born of the same age, you will observe that the female will first check. The female will first hold her head before the male. It is mark of strength and development. You also observe that if you have them of the same age growing together, the female beats the male child from when they start walking to age 10, 11, 12. Have you observed it? Have you observed it? If they are engaged in a fight, the woman is more aggressive and beats the male child. But the problem is emotion. The moment they get to the point of puberty, that the female realizes who she is and begins to get so emotional. Have you seen a female that grew up in the midst of men? Have you observed them? Very strong, strong-willed, very, very strong. That is woman's nature. But when the woman realizes from puberty the undulations, the development, that is going on, that will not attract men, that's when they get emotional. That's when they shrink. That's when they start losing their glory. And that is when man becomes stronger. Why am I mentioning this with you? I want to tell you that there shouldn't be any need for women to feel that she's a weaker sex at all. It's only in Africa, based on our culture. Our culture does not allow room for women. Women are only to be consulted, not to be allowed to be part of it. But the first major riot in Nigeria was led by who? Women. The Aba riot. When women said, we cannot pay tax. And they made a point. And we are stuck with it. So the problem we have is that the woman needs to rediscover who she is. Not because our culture has assumed that the woman is reduced to the Zaza room. And emotionally they get withdrawn. But I'm just telling you, I want to encourage you, just like our colleague said, that please, women should not consider themselves bitter sex. Women should take it as a challenge that yes, it is true. They say what a man can do, the woman can do better, but there's only one thing that both cannot do. Carrying a baby is a mark of strength. Do you know what it takes to carry a child, to carry an embryo for 9 months? A man will abort the child within 1 month. But it's a strength, both in character. The woman, sets her mind to accept that and carry it through 9 months. Men don't. So I want to encourage you women to please know that you are even going to be much stronger than man. All you do. But I don't say you should go and challenge a man. But know that you have the same endowment capacity to even do much better than men are doing. Thank you very much and welcome.
Yes, I've seen this. I saw this the first yes, I was supposed to be part of it, but I couldn't. I joined. I had a webinar. You not say anything. No, but it's I want to know more. That will help me very well. Yes.
The little contribution I want to make, or the little advice I want to give, is for us not to bury this knowledge that we have gotten here. Let us extend it to other younger and even elders in this academic profession or field. Like some of these our resource persons who are HODs in their various departments and in their various universities. They should write to the VC that there is a program like this that should be organized in their schools. And students and lecturers in those departments should be invited. If possible, all the resource persons here should equally be invited and be given the same topics. And maybe at the cost of trying to prepare for the seminar, they may equally go into more research, and from there others will come to know more about RNA. Thank you. That's a little contribution. Thank you.
That in the community, we must advocate for the inclusion of RNA
Yes, very important.
at the lower level. It's teaching time since none. And small things, big things, that's small. Let us not be small. That is one. Two, I am worried. Worried because the lady presenter was talking about discrimination. I wonder whether they are discriminated against or whether they believe themselves. Whether they discriminate against themselves. Because why I say that is that if you go to school today, you see that women have taken over every aspect of education. In the secondary schools in my state, I am from Anambra. Most of the male schools, there are fewer male. So most of the male schools are converted to co-education to increase the population. So you will rarely see a fully male school, boys school. The females are taking over the place. And in the education system, in the institute, in most of the institutions, they are there. If you check the nominal role of students, male against female, it's not that we are more female. They say that after school they will diminish themselves. I am worried. Because I don't think we are discriminating against girls. I think we are even
Sorry, I'm not playing. Somehow the men are responsible. Because I know we go to school, then along the line, so that there will be peace in the house. We obey. I have a very close friend who did have very, very intelligent. She did have BSc, came out with a good result. She got married. Whether with the husband to go for master, the man said over his dead body. The lady managed to get a form for PhD, and then started the PhD program. The day the man found out, he locked her out of the house and told her to go back to the parents' house. I hope you're following. And told her to go back to the parents' house. I'm making a point. The husband said, go back to your parents' house or you stop the program. And the woman said, to have peace in the house, she stopped. Because she had to go and call the people. They came back, and before the husband and the people, she promised not to continue with the program. Because she wants to develop herself, she has been sent out of the husband's house. Okay. So most of us who end up not coming out, it's not because we don't want to come out, but because they're not given the opportunity. This particular lady somehow, let me complete the story. This particular lady somehow managed to, get the PhD. Then to get a job, the man has opportunity to, the day the lady did her convocation, the man did not call. The man never supported her to go for the PhD. And do you know the reason? The man said, I am running my PhD. You have to wait for me to complete mine before you can go. This one is a complex.
There are two issues there. Number one issue, there are two issues there. You have addressed one. One issue is the issue of complex. There are some men who have inferiority complex. And so when they wouldn't want to say when they will get married, the woman will, according to them, outshine them. But they are very small in society. In the modern, I have done a research. And madam, this one may interest you. Before I go into my own, let me go into another system. The research was conducted in America. It included 500 female students, undergraduates. The basic question, the number one question is, why do you go to school? Why do you choose to go to the college? Why do you choose to go to college? If it's all, you say, why do you choose to go to university? You may not believe that 85% of females say they went to college because they want to get good husbands. Because they want to get good husbands. That's number one. I conduct my research. I want to know why younger generation women are struggling more than the men. And I want to see whether they do. And I realize that they do. Why? And I realize that younger men are more of gold diggers. They want successful women. That is why if a woman is ugly, has no hope of marrying, the man she gets the jobs in the bank, they will rush her to get married. So time has come when women will now have to do more, because the men or the boys are relaxing for them to do more. So the era of complex, when the men want to be ahead, when the men will not want their women to ascend them, has gone. In modern time, younger boys, all these younger boys you see, they are all gold diggers, looking for women who can take care of them. Already made women who can take care of them. So the women will have to, the younger generation have the opportunity of striving, even more than the men, even more than the male. But I don't think that the male is the male. I think the women, they need themselves.
Thank you, Prof.
I really appreciate what this program they organized. And I thank God that God should continue to give them strength and more wisdom to carry this program. And God bless.
I really want to appreciate our lecturer for the program which we had today. It indeed created a positive impact in me in particular, I don't know for others. And I'm really grateful for the guest speakers, especially Mr.
I'm very happy to be part of this seminar today. And most importantly, I want to congratulate our principal here, the rector of the Satec Polytechnic Enugu. He has been so helpful to some of us to excel in our academic careers. And also, I want to recognize my prof from our Faculty of Science at Ohio State University. He may not really capture my face, but you will see me. I'm also a researcher in computer science department. I'm on my PhD thesis now. Professor Ono is my supervisor. Ono, Ferguson. So, thank you so much. And God bless.
Thank you. That is the engineer in the house, Ishaka. I think you can see how diverse this session is. So, of COVID, everywhere we are all about molecular biology, therapeutics, drug design, and rest of them. That is for communicable disease. Then the issue of non-communicable disease also start rising. We also begin to see what is called epigenetic inheritance, whereby you begin, where people gather together to discuss prospect in RNA research. So when I started my MS, I was privileged to meet a scholar who wants to see people grow. I'm talking about Associate Professor Patrick Aja. So he treated me so well academically and research-wise. I felt happy, and during his birthday I decided to organize a webinar in honor of it. Fortunately, one of the participants, who is the HOD, did his postdoc, and fortunately also Associate Professor Aja did postdoctorate degree in Mbarara University. So he attended and become poised about the type of program I was able to organize to honor the birthday. It was actually a symposium to appreciate his work because he's a seasoned scholar. But then, this is Oga to him too. Then he called and said, can we begin this program in Africa? I said, yes, I can do it. So we were able to reach out through him to Associate Professor Kehinde of Liverpool John Moore University, who was his colleague in the lab or his supervisor during his postdoc. So they now give us the go-ahead, and here we are today. The first was the webinar, which was attended from people around the world, and it was successful. Then we decided to go physical and hybrid modes to integrate into it. The first in Africa was in Kampala International University. Then the communique is already in our website. The second is what is happening today, and first in Nigeria. So we are privileged to be part of it, and it's here. So just what I've said is in a brief. If you go to the website, Afri RNA, AFRILSC, you will read more. The journey is to make sure that young researchers take up research career development, to make sure that they contribute in what? Discovery of therapeutics in Africa. We should have those that should be making groundbreaking discoveries, and we are not different from the whites. Kehinde presented in Singapore on behalf of us. What he presented was what? Inclusivity in research, and they are now integrating it. So in our website, there is also space where we want to initiate collaboration and mentoring. How I grew was because of mentoring I got from BioClus organization in India. And by God's grace, research has not been a problem to me compared to other young researchers. So so that we won't waste time, this will be part of my own remark. I thank you all for being part of this program. One of the happiest things was the manner which those are invited or we invited have attended. Almost every of the presenters are as busy as we can't even talk of, but they make it to be here. The school management, I can't even imagine the welcoming hand that Professor Mike Okudri, director, gave me, together with the HOD, Dr. Ernest Eze, who has been part of my research journey over the years. In my early days, I'm an alumni of this school, an alumnus of this school, and also an alumnus of Ebonyi State University, where I'm currently doing my doctoral studies. But in my early days, the resilience in academics was instilled in me through the advice of Associate Professor Innocent Anidu. When I was the president of Student Academic Board, he will always say, if Peter is involved, I am involved. That alone made me to aspire to achieve higher, and here we are today. On behalf of the African Science RNA Salon team, I thank you for participating, and I want to tell you that the ball is in our court. We can make name for ourselves in Africa. Let us form collaborations. Let the younger researchers look up for a mentor. That is how to go about it. Let the advanced researchers give their time. We know you are busy. We know they are busy because administration disrupts academics. But then, to be promoted to the highest level, you have to take up administration position at some point. So we won't blame them, but you can do it for us. We need your time, and we also do what we call the post-conference engagement. It doesn't end here. You can reach out to me very soon if we get the support we want ahead to progress, because this is almost part of the last phase of this year's support. So if we are able to get the endorsement for that, we will form collaboration. Very soon we will start having institution chapters of RNA Salon. Thank you and God bless you. We acknowledge the fact that we can't afford to pay. So we call out our guest speakers. We call out our guest speakers. They have finished their meal. So we are calling first Associate Professor Ikechukwu Okore Igwe. So
Associate Professor Ikechukwu Okore. This is presented in the Mini-Symposium to you for being a contributor towards navigating RNA research career development and therapeutics. Thank you very much.
To come forward for award. Thank you.
African Life Science RNA Salon presents an award of honor to Dr. Innocent Chibuzo Anidu for guest frontier presentation in a mini-symposium being a contribution towards navigating RNA research, career development, and therapeutics. It's been given to you this day, 14th October 2023. Congratulations.
Present an award of honor to Dr. Antonia Kiruka Mba for guest/frontier presentation in a mini-symposium, being a contribution towards navigating RNA research, career development, and therapeutics. This award is presented this day, the 14th of October 2023. Congratulations.
African Life Science RNA Salon. This is an award to you, Dr. guest spiritual politician, National Museum, having symposium as a contributor towards navigating RNA research and development.
And Dr. Mrs. Kirumba to come and present certificate of participation to Professor Mike Okudele, Dr. Ernest Eze, and Sister Stella Remen. And Professor Achikano, you get ready to pick your certificate of. Even Prof.
This is from African Life Science RNA Salon, and this is certified that Professor Mike Okudele participated in the Mini-Symposium, not just participant, the chief host of this first in Nigeria, and I'm glad it is happening here. Congratulations. African Life Science held at our Savior Science Institute of Science and Technology on this day, 4th of October.
Participated in the Mini-Symposium of African Life Science RNA Salon held at our Savior Science and Agriculture and Technology in Nigeria on the 15th October 2023.
Participation at the African Life Science held at the SATEC in Nuku this day, 14th of October 2023. Thanks for coming to this particular event.
And Mr. Okolie, please, you will present the remaining certificates. When I call the person will come and collect and go. Please take this now. Thank you. Yes. Please, I have the Malo Lillian Chiamaka. Melo Lillian Chiamaka. I think I won't eat and forget myself. I will better.
I must talk. Look at God. This is our beloved son, in whom we are well pleased.
Ofo Chinedu Roland, come forward for the certificate. Okochi dị mma. Associate Professor Innocent and presents certificate of participation to Ole Jorom Oji.
That's my big brother. I'm my mentor.
I'm calling on Modern Solomon to come forward for certificate of participation. So we will give the privilege of leaving by some of the guest presenters who have a serious engagement and will be leaving now before we proceed with the certificate of participation and then closing prayer. Thank you for your time, Prof. Come and take your certificate. Followed by then Okorie Jennifer. Patrick Chigoziriugu to come for his certificate of participation. Please. Of all age, come forward for certificate of participation.
So, every other person come and use certificate as now and say you said right from you. Since your name is there, come and take photograph.
I can't live without a blessing. Thank you, sir. I'm very you see with us.
Next person. Okay, yeah. Please don't go. We have not said closing prayer. See, please say closing prayer. So, a closing remark.
Engaging symposium on leveraging RNA research for career development and therapeutics. I want to express my sincerest appreciation to all of you for your enthusiastic participation and thought-provoking insights throughout our deliberations. During this gathering, we had the privilege of listening to some of the brightest minds in RNA research who have been at the forefront of expanding our understanding of these fascinating molecules' potentials in the field of biomedicine and scientific innovation. With the growing interest and investment in RNA therapeutics and career development, we have heard about the exciting progress and opportunities that lie ahead in addressing some of the most challenges faced by scientific communities and patients worldwide. As we leave this symposium, let us take with us the insights, ideas, collaborations that have been fostered here. May they inspire us to continue the exploration and advancement of RNA research so that we can bring about a brighter future for the world and our future generations. Finally, I extend my warmest gratitude to our speakers for sharing their experience and expertise, our sponsors for their unwavering support, and our attendees for contributing to making this symposium an unforgettable success. We are also very grateful to the management and rector of the Satec Polytechnic Enugu for their immense love and contribution towards the success of this symposium. Thank you all, and may God bless you all.
Jesus' name. Thank you. May we share the grace. The grace of our Lord Jesus Christ, the love of God, and the sweet fellowship of the Holy Spirit.
So the students come for the program. So I will share feedback form to your WhatsApp. Please just help me and fill it. End it. Please, when you just fill it this night. We need to get the feedback this night for us to raise the communique.
they needed to carry office, every other item was constrained to them. Like say,
Anybody else with feedback? Extra sheet, ma'am.
Are you still around? Let me package my things. To use outside. Let me check inside. Bible hold it in the time. Okay, let's use the white. Let's use the white background.
Well, I know what to do.
I'm going, yes.
Back to work. Let's talk on phone.
Okay.
Okay. So what I need is your name, the role you play, that the impact you play on this program, your contribution, and what is your that means okay, that is all. Okay. Can I know you, sir?
Symposium that was organized at our Civil Institute of Science, Agriculture and Technology, THERAPEUTICS in Enugu, Nigeria, in Africa. The symposium was organized by African RNA Salon, and incidentally, a member of the RNA Salon that brought the issue of discussing RNA in Africa and presently now in Nigeria is a member of staff of my department, Mr. Peter Chinedu Ako. I've come to know him as a very astute researcher, and I was glad when he came to my office to announce the grant and that they were going to organize a symposium, that they have done the first in Uganda, and that the second to hold in Africa, in Nigeria, is going to be held in THERAPEUTICS in Enugu. I was elated and I thanked him. The only thing I did was to ask him if there are areas of interest and help that I will gladly do that. So I took him to the management and we offered him the necessary logistics that he needed to ensure that the program was a grand success. I'm particularly very grateful about the fact because the entire people that were invited all came around and a wonderful presentation was made. So I thank God for his care and his concern for all of us today, and I wish everybody that attended this program a safe journey back home. Incidentally, this RNA issue is a striking issue because it is the base for future medical treatment of diseases in the world. With this research, if it is well developed in Africa, Africa will very long go into solving so much of our problem as regards nutrition, as regards disease management, and all there is to keep good health. Thank you very much.
Thank you, sir. God bless you. Thank you.
Peter Chinedu. I'm a convener of this Mini-Symposium, and I'm also a founding member of African Life Science RNA Salon. So I'm resident here in Nigeria. I am the convener. I'm a doctoral scholar of Ebonyi State University, and I'm an assistant lecturer at our Civil Institute of Science, Agriculture and Technology. So it was a happy moment with researchers, and I'm happy I convened and people came. There was an insightful discussions, talks, and you can see we were able to achieve what we planned. That's why I'm very happy, and I know that RNA is being integrated in Africa. All thanks to RNA Society and Lexogene for giving us the opportunity to progress RNA research in Africa. I'm very happy, and I thank all participants for making out their time. Thank you.
Okay, thank you, sir. Thank you.
Thank you.